This lecture is going to cover the urinary system. This is going to be the first part of a two-part lecture. As we have done with our other systems, we're going to look at the who, the what, the where, the when, the why, the how of this system. We start with the gross anatomy of the urinary system. We talk about who are the major players. We look at the kidneys, the ureters, the bladder, and the urethra. The function is going to be to maintain homeostasis, and it does this by managing the volume and composition of fluid reservoirs, primarily the blood. The kidneys are going to be your go-to organs for monitoring, managing, maintaining blood volume, blood composition, etc. We can use this picture to guide us through and talk about all the different macrostructures of the urinary system. And we'll use a female in this picture as an example. So a couple things we want to point out as we identify some of these organs is going to be, well, number one, we see the kidney here, right? And we could say, well, how many do we have? And the answer would be 2. So we can number them. We have 1, 2. And the first thing somebody's going to say is, well, what's going on here? Why is one kidney higher than the other? And that's a great observation. And the answer is pretty simple. If you start adding some of our other organs in, and we think about what the largest organ, the second largest organ in our body is, and it occupies this space in the upper right quadrant, and we say the liver. So anatomically, because the liver occupies this space, when the kidneys develop, the right kidney had to be pushed down a little bit to accommodate that other organ. Now, dropping down from the kidney, if we follow this down, we have our left and right ureters. Again, we have them on both sides. Then we have our storage tank right here. This is our bladder. And the bladder is going to store amounts of urine, typically could be 400 to 800 milliliters, again, depending on the size of the male or female. And then from there, our urethra that is going to allow us to eliminate this waste product. Remember, we said that our kidney was a major player in regulating all things blood. So when we talk about the homeostatic functions of the kidney, we can isolate exactly what we are doing to blood other than filtering. So we are regulating blood ionic composition. So you say, well, which ions are we regulating? The first being sodium, the second being potassium, and the third being chloride. Aside from those ions, we also regulate blood pH. So if we think about a acid-base relationship, we should be thinking positive and negative. The positive ions would govern acidity, and the negative ions will cover bases or basicity. So we look at our two major portions, hydrogen ions and bicarbonate ions. And we think back to our previous lecture with respiratory when we talked about carrying oxygen and carbon dioxide, and we talked about how bicarbonate is moved and carried in venous blood as part of our carbon dioxide shift, and then the hydrogen shift as well, bouncing off that weak acid to also contribute to the acidity of blood. We're going to regulate blood volume. We do this by monitoring our water concentrations, okay? And we can adjust accordingly. And you see this throughout your daily life. If you wake up in the morning and you use the bathroom as one of the first things you do, and you notice your urine is pretty well concentrated, meaning it is a dark yellow. So we can say, well, what happened overnight? Our kidneys were still functioning, they were still filtering blood, still producing urine, but because you weren't eating or drinking or hydrating, your body went into a water conservation period, water conservation state, at which point your urine will become more concentrated to keep that interstitium, to keep those tissues well hydrated. And on the flip side, as you navigate through the day and you drink more water, more fluids, your body becomes well hydrated, and we can release more water. Our urine becomes more dilute and takes on a more clear or pale yellow appearance. So again, regulating water concentrations through blood volume. The next thing we're going to do is regulating blood pressure. This is going to be important. We'll look at a scenario later on. Remember, pretty much everything from here on out in this semester, when we talk about respiration, when we talk about formation of urine, we're dealing with pressure-driven systems, cardiovascular pressure-driven systems. So it makes sense, since the kidneys are dealing with a massive amount of blood every day, that they are the go-to player for regulating blood pressure. We can now look at the homeostatic functions of the kidney. Number one is going to be maintenance of blood osmolarity. It does this by monitoring, maintaining fluid balance, electrolyte balance within the body, within the tissues, and within the interstitium. The second function is going to be production of hormones. Two hormones are produced here. The first one we're going to talk about is erythropoietin, or EPO. EPO is a glycoprotein. Its function is to act on bone marrow, and by acting on bone marrow, it's going to increase the production of red blood cells. So the question is why? Why would this happen? When would this happen? Well, as usual, we respond to a stimuli. The stimuli that will increase EPO production from the kidneys would be bleeding, or if you move to somewhere that is of high altitude where oxygen is scarcer, it's going to trigger the release of EPO. The second hormone we talk about being produced here is calcitriol or calcitrol. Calcitriol is vitamin D3. It's the active form of vitamin D. We can talk about where it's made and how it acts, but let's look at the first action of calcitriol. Calcitriol's main action is going to communicate with the cells of the intestines, and what it does is it's going to promote the absorption of calcium and phosphate from food. From there, it's going to move to the bones to mobilize calcium, and from bone to blood. The third thing the kidneys do is excretion of metabolic wastes and foreign substances. This can include drugs or toxins. We talk about metabolic waste. The kidneys are primarily involved in removing nitrogen-containing wastes, and it does this to prevent toxic buildup. Well, you say, well, where does this metabolic waste come from? Well, the metabolic waste occurs during breakdown of nitrogen-containing proteins. This metabolism also involves the removal of nitrogen from amino acids, and amino nitrogen is often removed as ammonia. So we go back to protein degradation. We go to digestive system that's breaking down protein, breaking down the amino acids as ammonia, storing it in the liver, and then converting it to urea. So from there, that urea is going to be safely removed from our body as waste, and the kidneys are going to be the major player in that. The last thing the kidneys are going to be doing is regulation of blood glucose levels. Now, sugar is a smaller compound. It does make its way through those fenestrated capillaries inside the kidneys, inside Bowman's capsule of the glomerulus, and it will get re-picked up by our blood once it's in the filtrate. The problem occurs when we have clinical conditions like diabetes, where way too much sugar is in the blood. The kidneys can't possibly reabsorb all that glucose, and as a result, it gets excreted in our urine. So when we do urinalysis and we see high levels of sugar, this could indicate a host of issues, number one being diabetes. Renal anatomy. We're going to examine exactly where the kidneys are located, in what cavity, and how they are protected. So the kidneys are retroperitoneal, so retro meaning behind, peritoneal meaning behind the peritoneum. And we're going to see that half of the kidneys are going to be partly protected by the lower ribs. This is an important bony structure that can protect the upper half of these kidneys. If you look on the left-hand side over here, we have our different layers. We go from superficial edema. We'll relook at this again in its own private slide. But we have our renal fascia, which is a grouping of collagen fibers of fibrous connective tissue, anchoring these kidneys in place, kind of like Spider-Man came by and shot a web and stuck these to the back of your body. An adipose capsule, so we have one adipose capsule number 2, followed by a renal capsule number 3, which is going to be continuous and surrounding the ureters. And we have our right kidney followed by, again, since we have 2, our left kidney. We can take a different cut and look at a parasagittal plane, and we can really see rib 12 here coming down, protecting a portion of that kidney. This is our right kidney. Our layers again, we can count them as we go to the renal fascia right here. And then we have our fat or adipose layer, and then our renal capsule. When we look at the kidney, we'll look at the renal capsule right there. We look at renal anatomy, so the entire kidney as a whole. Remember, like we saw with the lungs, we have an indented area right here called the renal hilum. This is going to be an entrance for tubular structures like the ureter, blood vessels, the renal artery, the renal vein, nerves, and lymphatics. We can go back to those external layers, look at that connective tissue. Again, bundles of collagen fibers, dense, fibrous connective tissue that's going to form that renal fascia that's going to anchor those kidneys down with the adrenal glands on top and anchor them to other structures. We have a fat capsule. Again, that is an adipose capsule that's going to help protect as well as anchor. And then lastly, a renal capsule that is going to surround the entire kidney, and it's going to be continuous with ureters. If we look at internal renal anatomy, it brings us to the divisions. We go from large to small. We start with, and like every other capsular structure we looked at when we looked at the adrenal gland in endocrine, when we did the spleen, in the lymphatic system, we deal with an outer layer and an inner layer. We deal with the renal cortex being the outer layer, right here, renal cortex, outer layer, and our inner renal medulla, so the medullary layer in the inner portion. We're going to have renal pyramids. This is going to group together to form the renal medulla. It's going to be our secreting apparatus, and this is where we're going to find our tubules, right? This functional unit, the nephron of our kidneys. We're going to have extensions of the renal cortex that goes in between the renal pyramids. They're called the renal columns, and they're going to help right here function, anchor that cortex in place. So we can look at this two ways. We're going to focus more on the picture on the left to look at some of this internal anatomy. And we can start on the outside. Right here, this dark purple that you see is going to be our renal capsule. We can now bracket this region right here. This is our cortex. We look at the extensions in between those renal pyramids where that cortex extends, forming our renal columns. Now, every pyramid that you see here, 1, 2, 3, 4, right? These renal pyramids are going to have a base here and an apex at the top. The apex is going to extend into our series of funnels. We'll talk about that in a second. So for our renal medulla, we have our renal pyramids, our renal columns, and our renal papilla. So again, papilla projection. Now that we have our first set of funnel open, this whitened area right here is going to be our renal papilla. From there, we enter into a series of funnels from small funnel to large funnel. This is where we're going to direct the path of urine and where it's going to drain into the ureters, into the bladder. So we start at our first funnel. So before we outline our funnels, let's go ahead and erase all the marks that we've done so far so we get a nice clear picture. And we start right here. This is our first set of funnels, smallest funnel. It is known as the minor calyx. Now, all these 1, 2, 3 minor calyces are going to drain into this larger funnel. So let's erase. Let's outline that larger funnel. That larger funnel is a major calyx. From there, it's going to drain into this larger region. So notice we went from small to larger to largest. This is our funnel system, forming our renal pelvis. Once in that renal pelvis, we're going to take that urine and we're going to drain it right down into the ureters, and it's going to pool inside the urinary bladder. Blood and nerve supply to the kidneys play an important role. Number one, we look at blood supply. When we look at the size of the kidneys, they make up less than 0.5% of our total body. But remember, we said that they are massive players in blood volume recognition and regulation. So they receive 20 to 25% of your resting cardiac output. That's a massive amount of blood. Our nerve supply, we talk about the kidneys' nerves primarily carrying sympathetic outflow. And we say, well, why this sympathetic system? So we deal with fight or flight and its influences on the renal regulation of arterial pressure and specifically body fluid composition. So we see that sympathetic nerves are going to mediate changes in the urinary sodium and water excretion, and it does this by regulating the renal tubular water and sodium reabsorption throughout the nephron. So ultimately, changes in renal blood flow and the glomerular filtration rate are regulated by constriction of all those arteries and veins, specifically the arteries, that renal vasculature, as well as changes in the activity of that RAA, that renin-angiotensin-aldosterone system. It does this by regulating the release of renin from the juxtaglomerular cells. These are all points that we're going to discuss further on. But from a general perspective, realize that we're dealing with sympathetic outflow and they're going to be regulating. So on that arterial end, they're regulating blood flow through the kidneys. Blood supply through the kidney and the nephron. So if we're going to talk about how blood moves from to the kidney, through the kidney, and then back out, we're going to start right here. We'll mark it number 1, our renal artery. So blood's going to come in, it's going to pass through the hilum, and it's going to reach this portion right here. Mark it number 2. This is going to be our segmental artery. From there, we're going to go in between, we'll mark it 3, in between our renal pyramids and our renal column, and we're going to call this our interlobar artery. From there, we're going to go to the base of the pyramid. This artery is going to be called arcuate. And then we're going to radiate into the cortex, calling it our cortical radiate. From there, we're going to loop all the way up, just to zoom on in to that nephron. We're going to come in, so we pick up right here from the cortical radiates, and we're going to come in, so if we label 4 here for our arcuate, 5 cortical radiate, 6, we come into our afferent arteriole. From there, we go into the glomerulus, and the glomerulus is going to filter out the plasma, the blood, filter the blood. And then we go to number 7, to our efferent arteriole. Now, this is where it gets a little bit tricky because we're going to go in between the tubules. Now, if we have a juxtamedullary nephron, which have longer loops, it's going to have something called, if you see right here, the vasa recta. If we have cortical nephrons, we're going to go right to peritubular capillaries, okay? So if we look on our list here, we go efferent arteriole, then to peritubular capillaries. So from those peritubular capillaries, we're going to switch over to venous flow, and we see that we have our peritubular veins, or venules, and we'll find those right up here. Now, from our peritubular venules, we're going to now reverse back down. We're going to have our cortical radiate vein. So if we come back, and we're going to switch it over to white, and we'll pick up right here our cortical radiate vein. Remember, we're flowing this way. From our cortical radiate vein, we're going to hit our arcuate vein. From our arcuate, we're going to go in between the pyramids, our interlobar. And from our interlobar, there is no segmental. We go directly out to our renal vein. Now we could talk about the structures of that nephron that we just got looking at the blood flowing through it. So we can start right here at the renal corpuscle. So we'll check it. The renal corpuscle is going to be comprised of 2 major structures, a glomerular capsule and a glomerulus. So that glomerular capsule is similar to our serous membranes, where it has two layers, outer parietal layer and an inner visceral layer. And it's going to set up a capsular space. And that space is going to be important for allowing the filtrate of what's been filtered from the blood to move through and into the tubules. Now, once we take care of this structure, the renal corpuscle, filtrate is going to flow this way. And we're going to hit a couple different regions. We think about squiggly. Anytime we have a tube that squiggles, we're going to call it convoluted. Okay? And we have two squiggly tubules. We have one here and one here. Now we just name these tubules based on where they are in relation to the pole of this nephron or the renal corpuscle. And we're going to use words kind of like we used when we were naming bones and landmarks on our appendages, words like proximal and distal. So we go to this first squiggly tubule, and it's close to the pole or close to that renal corpuscle. So we're going to call it the proximal convoluted tubule. And then we're going to move down, we're going to travel down, and we're going to hit our nephron loop, or loop of Henle. And they're pretty much named just on the direction of the filtrate. We have the descending limb, and as we hit that loop and we go up, we have the ascending limb. And then we hit our next squiggly tubule that's going to take us to further away from the nephron, and it's called our distal convoluted tubule. Now, again, if we assume that this is a straight line, where it really, in real life, it really isn't, and I'm going to erase a lot of this stuff because I want to go back and point something out. With all my markings, you can't see it anymore. So in real life, when we get to the actual orientation of this loop, this distal convoluted tubule actually turns back around this way. And if you see this V formed right here from the afferent and efferent arteriole, this portion of the distal convoluted tubule and the ascending limb is going to move back between this V. So if we think of it like this, we're putting the end of this system back at the beginning. And that's a little weird to think about, to say, well, why would we put the end of this structure? And if we go back to the nephron and what the function of the nephron is, we're essentially making urine, right? We're absorbing, reabsorbing, secreting, we're concentrating, diluting, we're making urine or waste product and urine to be released out of the body. So why at the end are we putting this back to the beginning? And it's essentially a checks and balance system. We're checking the end result so we know what to do at the beginning. Now, we're going to elaborate on this a little later, but keep that in the back of your mind. Now we can examine each separate structure in of its own and talk about specifically what they're responsible for doing. So we're going to focus in on the renal corpuscle. And as we said before, the renal corpuscle is going to be made up of two major parts. The first is the glomerulus. Now, I want you to think back to AP1 when we looked at our epithelium. And we talked about simple squamous epithelium. And if you were in my lab, we talked about a ball and a ball. And we looked for this big ball that was surrounded by this thin little ball. Well, that's what we're talking about here. We're talking about those two balls. That is the renal corpuscle. The outer ball is Bowman's capsule, right? It is this visceral layer of podocytes, which is going to wrap around the capillaries. Basically, what it's doing is it's forming a filtration barrier. Now, the filtrate is going to be collected in that space between the visceral and the parietal layers. Now, if we go inside to that inner ball, this is the glomerulus. It's going to be a mass of capillaries. And again, if you go back to the capillary lecture that we talked about these fenestrated capillaries, and the example we used was inside the kidneys, inside the renal corpuscle. So the glomerulus is going to be a mass of fenestrated capillaries. It's going to be fed by that afferent arteriole that we just outlined. It's going to serve its function as to filter the blood, and it's going to drain into the efferent arteriole. We can zoom on in and really take a look at this renal corpuscle, and we can see a couple things. If we go up to blood supply and we look at blood coming in, a couple things to notice. Look how thick and wide and fat that afferent arteriole is versus the efferent arteriole, how thin it is. And this is going to be important as far as regulating the diameter of that arteriole and controlling the blood pressure and the amount of blood coming in. We can go to the outside. Look, there's our simple squamous epithelium, that parietal layer of Bowman's capsule, capsule. We have the capsular space right here. And then we can see this yellow, these podocytes. You can see that little nucleus, that cell with all their foot-like projections, that visceral layer, that modified visceral layer that are forming these slit-like projections surrounding that fenestrated capillary. And again, we're going to call them, from those podocytes, to our pedicels. If we look at the picture in the lower right-hand corner, here's our ball in a ball. We have our outer ball and our inner ball, right? So our simple squamous epithelium with our fenestrated capillary in the center. Now, one more thing I want to point out in this picture is this tube-like structure right here. This is going to be the ascending limb of the nephron loop, could also be the distal convoluted tubule, okay? And notice that it comes back around and moves between that V-like structure of the afferent and efferent arteriole. Now, this is important. So we already alluded to this. We said that we're putting the end back at the beginning, okay? So let's look at its anatomy. We have some modified simple columnar cells, these elongated cells called the macula densa. And then we're going to get even further modification of cells called mesangial cells. So again, just introducing the terminology, and we're going to look in a little bit exactly what these structures and cells do with regards to contracting, with regards to secreting things, etc. Now, we can look at the renal corpuscle again and look at the glomerular endothelial cells and see that they have those large fenestrations, those pores. Those pores, when you go back to our capillary lecture, were leaky. It was allowing for, size-restricted stuff to move in or block and essentially filter. Between there is this basal lamina that's going to lie between that endothelium and the podocytes. And again, it's going to help us filter some more, and we'll look exactly what these layers are filtering. From there, we have our podocytes with their foot-like pedicel projections, between which are going to be our filtration slits. Now we can look at it in depth at a picture. Look at these pores. So we see that fenestrated capillary right here. We see all these pores. This is of that endothelial cell line. Its job is going to be prevent filtration of blood cells, so red blood cells, white blood cells, but it's going to let pretty much all the components of blood plasma to pass through. But luckily, we have this blue basement membrane here. That basement membrane is going to help prevent filtration of larger proteins like albumins. Iron globulins are not going to make it through, are going to be blocked right here and maintained within the blood. And then we go out even further and we have our slit membrane right here between our pedicels. So here's one pedicel, another pedicel, again those foot-like projections off of our podocyte that's right here, okay? Now those pedicels are going to further prevent filtration of medium-sized proteins. So again, we're blocking the larger proteins and even the medium-sized proteins. Again, trying to keep the pressure within the blood with all those large components. Two kinds of nephrons. We mentioned before when we looked at our blood supply that we have two different types of nephrons. So we're going to discuss them and then we're going to show you a picture to kind of explain exactly what they look like. So the most numerous of our nephrons are called cortical nephrons. About 80 to 85% of the nephrons in each kidney are cortical. We can look at the total number of nephrons, and we can say that the average person has anywhere from 1 to 1.5 million nephrons per kidney. So again, the majority of these are going to be cortical. And if we look at the name cortical, it kind of tells us where we find it. So cortical sounds like cortex. So we're going to see that the majority of this nephron is going to lie in the cortex, the outer portion of the kidney, which means that its loops of Henle are going to be really, really short, and they're only going to extend into the outer region of the medulla. Now, the job of these cortical nephrons is going to be to create urine that's going to have an osmolarity similar to that of blood. Now, we look at the next slide and we say, well, what can we see in this picture? Now, we look at the nephron. We got a lot of blood vessels intertwining here, and this is going to be important. Now, we see our arcuate artery right down here. We have our cortical radiate going up. And now we go into the actual nephron. And we see that the majority of the nephron, if we look right here, the majority of that nephron is going to lie in the cortex, and only a small percentage is going to lie in the upper margins of the medulla. Now pay note to these capillaries. Look at them. They're sweeping, they're swirling, they're twirling around the tubule. Okay? Remember that the jobs of these nephrons is to make urine that has a similar osmolarity to that of blood. So what's going on here? Okay, well, we're going to look at the secretion and reabsorption routes, and we're going to see that these peritubular capillaries, remember, we're not vasa recta, that's only in the juxtamedullary nephron. Here we're in the cortical nephron, we have these peritubular capillaries that are going to, peri meaning around, surround the tubule and lead into our venule capillaries. So if we look at flow of fluid through a cortical nephron, we go to our 1, Bowman's capsule or glomerular capsule, 2, the proximal convoluted tubule. We come down the descending limb, back up the ascending limb, and then we hit the distal convoluted tubule. Okay? Now let's look. I want you to pay note again that when we come up, that distal convoluted tubule, that ascending limb, is moving between the afferent and efferent arteriole. Again, I keep trying to drill this in because we're going to look back at that, and I want you to draw back on this as we look at the function of that structure. Now we come into these collecting ducts, and from the collecting ducts, we're going to enter into the renal papilla. If you go back to the renal pyramid, that white portion at the bottom, and we're going to slowly drip urine into the first of our smallest funnels, the minor calyx. The second nephron we find is a juxtamedullary nephron. Now let's look at its name, kind of like we did with the cortical nephron. Juxta means next to, medullary means medulla. So this name tells us where we find these nephrons. The majority of these nephrons are going to be located in the medulla of the kidney, which means that they're going to have these really, really long nephron loops. Okay, so the renal corpuscle is going to be deep in the cortex with these long nephron loops. We're going to receive blood from peritubular capillaries and a vasa recta. Now in the next picture, we're going to see what that vasa recta looks like, because it looks completely different from those peritubular capillaries surrounding those descending and ascending limb in the loop of Henle that we saw in our cortical nephrons. Our ascending limb is going to have thick and thin regions, and we're going to enable the kidney to secrete very concentrated urine. So the function of these juxtamedullary nephrons is really to create concentrated urine and fine-tune that urine output. So let's take a look at what this structure looks like. And right away, we see a small part in the cortex, and the majority of this nephron is going to be located in the medulla. Now pay note to these vasa recta, long, sweeping capillaries. Okay, this is going to help set up something called a countercurrent exchange mechanism. So we'll talk about that a little later on in this lecture. and also in our next part 2 of this lecture, and what that does to form a concentrated or dilute urine. Now again, we look at the flow of fluid through a juxtamedullary nephron, pretty much the same as our cortical. We have 1 Bowman's capsule, 2 proximal convoluted tubule, 3 descending limb, 4 ascending limb, 5 we hit that thick ascending limb of the nephron loop, and then 6 we go to the distal convoluted tubule. So then the question is, why are we changing? Let's erase some of these structures here and see what is going on. So I want to point out, before we get into too much detail, because we will look at this a little bit later, what's going on here. So if you see closely, let's zoom on into this structure. If you see that right here, we're going from thick to thin. And we say, well, what's happening here? Why are we switching over? And we can think about this in a common sense fashion. Right? Let's think of it logically. When the tube is thinner, what will be happening? Well, in a thinner tube, you would think that we're allowing things to move in and out pretty readily. Well, what's going on with this thicker tube? Well, if we make a thicker tube, we're trying to block stuff from moving out. Well, in this ascending portion, we are trying to block something. We're going to specifically block water. So the reason why we block water here is we're trying to maintain the dilution of the urine. And again, when we look at urine formation, we'll talk about the glycoprotein that is lining that tube, that is preventing water from moving out, and how important that is for maintaining a countercurrent exchange mechanism. That brings us to one of the last anatomical structures we're talking about, and that is the juxtaglomerular apparatus. So let's look at our words again. Juxtaglomerular, next to the glomerulus. So let's think back to what we just said. What structure is coming back up in between the afferent and efferent arteriole to form a complex called the juxtaglomerular apparatus, or complex? Well, it was that ascending loop, that ascending limb, that distal convoluted tubule. It's going to contact the afferent arteriole at a structure where we saw those modified elongated columnar epithelial cells called the macula densa. The walls of that arteriole is going to contain smooth muscle cells. These are called juxtaglomerular cells. Those juxtaglomerular cells are going to be able to regulate blood pressure in the kidney. And it's going to work in conjunction with the autonomic nervous system. So we say, why? Why do we need to regulate blood pressure? Well, there will be certain scenarios where, say, blood pressure is low. Remember, these are pressure-driven systems. So if blood pressure is low, so we can draw blood pressure. If blood pressure is low, that means that the amount of filtrate that's going to be created, so filtrate created is going to be also lower. So in order to increase the amount of filtrate, the amount of blood that we're filtering and the filtrate being produced, we need to increase this pressure. How are we going to do that? Well, there's a series of ways that we can, but number one, we're going to allow for this apparatus, these cells, to contract down. Those mesangial cells are going to contract down and make the lumen of those vessels much smaller, while at the same time artificially increasing blood pressure and increasing filtrate production. Renal physiology, looking and examining urine formation. Now we're going to start quite simply with this and just look at some basic routes and processes, and then we're going to get deep into the actual reabsorption and secretion routes. What's being reabsorbed, what's being secreted, where it's being secreted and reabsorbed, and how. So we look at a couple things here, and this picture does a pretty good job to show us the excretion of a solute. So how do we get that? We're going to get glomerular filtration plus secretion minus reabsorption. So look at each of these processes below. We start with number one, glomerular filtration. blood is moving into and through that fenestrated capillary, through the glomerulus, out the efferent arteriole. What's happening here? Inside that glomerulus, blood plasma and dissolved substances, anything smaller than most proteins, sugar, some electrolytes, things like that, are going to get filtered into the glomerular capsule. And it's going to move this way. Once it moves in that capsule, it's going to start moving through the renal tubule. Now what happens? We get two processes. We get this right here, it's number 2 in our process, which is tubular reabsorption. And basically what's happening here is all along the renal tubule and the collecting duct, water ions and other substances are going to get reabsorbed from that renal tubule, the lumen of that renal tubule, and put back into the peritubular capillaries and ultimately back into blood. Now that's important. We have to take that water. Now this part right here at that first portion at our proximal convoluted tubule, we're going to be taking a large percentage back of that water that was filtered out from step 1. So a lot of plasma, a lot of water was filtered out, and then we're going to reabsorb it back. We're going to follow a couple processes here. This is going to be called obligatory water reabsorption. So if you think about the terminology, obligatory, right? We're going to write this down. We're going to look at this again a little later. Obligatory water reabsorption. So here, obligatory means we are obligated to reabsorb water. And this is to make sure that we keep blood volume pretty consistent and therefore blood pressure pretty consistent. The third process we see is right here. So notice in step 2, we are going into the blood capillary. Step 3, we are secreting from the capillary and putting it back into the renal tubule. So we say what's being pushed out of this blood vessel and sucked back into the renal tubule? Well, along the renal tubule and that collecting duct, we're going to see waste products, drugs, and any excess ions that get secreted from the peritubular capillaries going to be put back in, secreted back into the renal tubule. Ultimately, these substances are going to make their way into our urine and we're going to excrete them. Glomerular filtration. We're going to continue by looking at all the forces that affect the amount of filtrate and what's being filtered through that glomerulus. Right off the bat, look at the first bullet. We are driven by blood pressure. Remember, we've been talking about this and I've been hinting to this again and again. These are pressure-driven systems. If our blood pressure is too low or if our blood pressure is too high, this can directly affect glomerular filtration rate, or GFR. Okay, so some of the forces that are working with and against glomerular filtration is we're going to see that blood pressure is going to be opposed by a couple things. Number 1, capsular hydrostatic pressure. So the water pressure from the capsule is going to exert a pressure back onto the glomerulus. And then we have this blood colloidal osmotic pressure. Again, remember, blood colloid is those large components of blood that is exerting an osmotic pressure as well on that glomerulus. Remember that we are going to allow for water and small molecules to move out of the glomerulus. And we say how much, because we alluded before that blood or the kidneys were a major player in focusing on blood volume, on working through blood issues and blood composition. And we said that it filters about 20 to 25% of our resting cardiac, or receives 20 to 25% of our resting cardiac output at any given time. So if we look in one day, look at the amount of water that's passing out and into the glomerular capsule. We're dealing with 150 to 180 liters. This is a massive amount of filtrate being created in a day through your kidneys. So your kidneys really are workhorses. They are pumping through massive amounts of blood, creating 150 to 180 liters of water that's moving through this system in any given day. We can look at glomerular filtration a little more and look at the amount of filtrate that's formed by both kidneys each minute. We're going to call this glomerular filtration rate. So homeostasis is going to require the kidneys to maintain a relatively constant GFR. Here's the problem. If GFR, glomerular filtration rate, is too high, substances pass too quickly and are not able to be reabsorbed. Here's the analogy. I go back to an episode of I Love Lucy that I saw when I was a child. Lucy worked in a chocolate factory. Her job was pretty simple. It was to take the chocolates coming past her on the conveyor belt, put them and package them in a box, close the box, and continue with her job. The problem that happened in this episode was that conveyor belt got faster and faster and faster. As the conveyor belt got a little faster, Lucy had a hard time putting those chocolates back into the box. She started only putting, instead of the 8 that she was getting in there originally, she went down to 5. She took the extras by putting them in her mouth. Then the conveyor belt got even faster, and she was able to get almost no chocolates into that box. So she started throwing them over her shoulder, shoving them down her shirt, and putting whatever she could in her mouth. So this is a good representation of what happens when blood pressure gets too high. Those substances pass too quickly, and just like I Love Lucy, or just like Lucy was not able to keep up with that high speed and put the chocolates in the box, your kidneys cannot keep up with that high speed and reabsorb components that they need to adequately. Now, if it's too slow, now imagine if that conveyor belt was too slow. Lucy would be sitting there tapping her foot, waiting for more chocolate, and she would be filling those boxes perfectly. She may even have enough time to put a bow and wrap it up nice and neat. So this is the same thing in our kidneys. If things move too slowly, we're going to see that nearly all of the components of that filtrate gets reabsorbed. And that's bad, because that means that some of our waste products are not being excreted. And as a result, we're keeping that back, all those waste products, reabsorbing it back into our blood, back into our body. So not a good scenario. So this is kind of like that scenario where moving away from Lucy and into kind of the Three Bears, where the porridge was not too hot, not too cold, but just right. That's where our GFR needs to be. It needs to be right in the middle at a perfect, consistent, constant pressure to maintain a relatively constant GFR. We can put GFR to number and to a calculation. So, kind of like what we saw in the capillaries where we talked about net filtration pressure, we're going to do the same thing here. So we deal with NFP, so right here, net filtration pressure, and we're going to take all our components of the system. So we're going to deal with blood coming in. We're going to call this glomerular blood hydrostatic pressure. So number 1, glomerular blood hydrostatic pressure. We're going to put that at 55 millimeters of mercury. Now this is what's coming in. We're going to have to subtract all of our external forces. See these purple and blue arrows that are moving down and exerting a force onto that glomerulus. So the first is that capsular hydrostatic pressure, and that capsular hydrostatic pressure is going to be the pressure of essentially what the filtrate inside this space, so all the filtrate inside this space, the capsule is going to push down on that filtrate, which is going to put a pressure onto that glomerulus. We're going to put a number to that. It's measured at about 15 millimeters of mercury. Now we deal with blood colloidal osmotic pressure. Again, we're talking about the larger components of this filtrate is going to exert a pressure back onto the glomerulus, and that's going to give us a pressure of about 30 millimeters of mercury. So now let's zoom back down and we'll talk about our math. So we take our blood coming in, the pressure of the blood coming in, we subtract it from our CHP, capsular hydrostatic pressure, is 15. We subtract that from our blood colloidal osmotic pressure, which is 30. It's going to get us a net filtration pressure of plus 10 millimeters of mercury. So that's good. Remember, we're moving stuff from a high pressure to a low pressure. So that means net filtration is going to be promoted in an outward flow. So we'll keep that green arrow. We'll make it a little bit bigger. So NFP is outward. Good. Now, when can this become skewed? Let's look at blood pressure. Say we're bleeding internally or we're bleeding externally and we have some massive blood loss. If this number here drops down to, say, let's pick 45 millimeters of mercury. Now we're even Steven, and we have 0 millimeters of mercury on the NFP side, net filtration side. So what happens? We have none of this. No net filtration, which means that urine output goes down. And this can be measured by the amount of urine that's being created. Okay? So one quick scenario. We will look at other scenarios in a little bit, but I just want to show you what happens when these numbers go awry. Well, let's look at one other one. Let's bring this way high up. Let's say our blood pressure is too high, and we go to 75 millimeters of mercury. Now we subtract 45. Now we are plus 30. So we'll draw our 30 right here. Now look, that means that our net filtration pressure is plus 30. We're going to, well, let's erase this right here. And we'll redraw it with our big, monstrous blue arrow. So that means that our net filtration pressure is really high. Here's our I Love Lucy scenario. This stuff is going to be flying by these cells, not allowing all these microvilli on these cells to do their function, which is reabsorb. Let's take a look at some GFR, some glomerular filtration rates. So for averages, in males, the average GFR is about 125 mils per minute for, again, the average male. For females, our GFR is about 105 mils per minute. And then we say, well, what really controls this? Because again, we go back to pressure too high, pressure too low. What can control the amount of filtrate coming through those tubules at any given time? We can lower this GFR or increase this GFR. So it's going to be controlled by three things. One, renal autoregulation. So the kidney has the ability to autoregulate itself, so can control the blood pressure, either increasing it or decreasing it to artificially increase or decrease GFR. Secondly, we have neuroregulation. So the central nervous system, the autonomic nervous system, plays a role here in controlling the GFR, as well as hormonal regulation. We talk about catecholamines, we talk about adrenaline, norepinephrine, epinephrine, and other hormones that are going to increase heart rate, increase blood pressure, and ultimately increasing or decreasing glomerular filtration rate. Continuing with renal autoregulation, we're going to look at a couple mechanisms that's going to help allow the kidneys to self-regulate. The first is a myogenic mechanism. Now we look at this word here. We always look at prefixes and suffixes. Myo, muscle. So we're dealing with smooth muscle cells that are located within the afferent arteriole. Now if you go back to a couple slides where I outlined the thickness in that afferent arteriole. So we had that, I'll see if I can draw it real quick for you. We had the afferent arteriole coming in and it was thick-walled, right? Whereas the efferent arteriole coming out was much thinner, okay? So that thick wall was lined with smooth muscle. So that smooth muscle is going to be, so let's draw an arrow coming in, and then we hit the glomerulus here, and then we come out. So those smooth muscle cells in the afferent artery are going to contract in response to elevated blood pressure. So exactly how do they do that? So increased blood pressure increases the tension in the vascular wall. And as a result, this thickened right here, that thickened smooth muscle, that vascular smooth muscle is going to contract. Now on the flip side, when a decreasing in blood pressure is detected, so decreased blood pressure decreases the tension and the smooth muscle relaxes. So, overall, to make this a little simple, when blood pressure increases, the size of this lumen decreases as we contract on this wall. When blood pressure is lower, so when a decrease in blood pressure is detected, the lumen gets bigger and the wall relaxes, moving outward. Now, let's go ahead and delete this drawing because we're going to talk about the second pathway for renal autoregulation. I don't want that drawing to convolute anything. So we talk about tubuloglomerular feedback. So the feedback between the tubes and the glomerulus. Now, what's happening? We already said that high glomerular filtration rate is going to diminish reabsorption of some of those solutes, and that could be bad. The macula densa is going to use the composition of the tubular fluid as an indicator of GFR. So what it does is actually sense either a large sodium chloride concentration or a low sodium chloride concentration. If GFR is high, it's detecting a high sodium chloride concentration, which is going to indicate a high GFR. So, too high sodium concentration detected by the macula densa. As a result, it's telling itself that it's detecting too high a GFR. We're losing too much into the filtrate. It's going to inhibit the release of nitric oxide. If you remember, nitric oxide's a major vasodilator. So by doing this, by inhibiting nitric oxide, we are essentially allowing for the afferent arteriole to constrict, lowering the amount of blood and blood pressure entering the glomerulus, ultimately lowering GFR, bringing it back into a normal range. That brings us to a complete cycle of renal autoregulation. We can look at this negative feedback loop to see exactly what's happening. We have a stimulus, number one, that is disrupting homeostasis by increasing our glomerular filtration rate. As a result, the macula densa, so now we're here, the macula densa, these are the cells of the juxtaglomerular apparatus, are detecting those high sodium chloride concentrations. So we see right here, sodium chloride, high concentration of sodium chloride in water. And that JGA is going to be the control center. It's going to affect the diameter of the afferent arteriole by constricting it, which is going to decrease blood flow through the glomerulus, which is going to decrease GFR. And we're going to maintain this process until GFR is back into normal range. When it hits that normal 125 mils per minute for males, 105 mils per minute for females, it will negate, shut down this feedback loop. Neural regulation. So first and foremost, the kidneys are going to be supplied richly by sympathetic fibers. So we discussed about the autonomic nervous system and how the sympathetic portion is directly communicating with the kidneys. When we talk about release, we talk about norepinephrine causing vasoconstriction within the kidneys, and it's especially going to affect that afferent arteriole. If we go back to that smooth muscles, the thickness in that afferent arteriole that we covered in our past few slides, this is where that neuroregulation is really going to take hold. We can look at some situations such as exercise or hemorrhaging. This would be strong stimulation. And what predominates is going to be constriction of that afferent arteriole. Overall, urine output's going to be reduced and more blood is going to be available for other organs. Again, if you think about fight or flight, we're diverting blood nutrients to our muscles, to other parts of our bodies, away from our rest and digest organs, which would be urinary, digestive, etc., so that we can sustain a fight or flight scenario. Hormonal regulation. We've introduced before the renin-angiotensin-aldosterone system. Our next slide is going to go through that. But if we look at a couple hormones that are regulating or working with glomerular filtration rate, angiotensin 2 is going to be one of them. It's ultimately going to cause constricting of the afferent and efferent arterioles, so we're constricting both the blood coming into the glomerulus and blood leaving the glomerulus, overall decreasing blood flow, overall decreasing glomerular filtration rate, GFR. Atrial natriuretic peptide, ANP. The job of ANP is going to be to relax those mesangial cells, ultimately increasing capillary surface areas and increasing glomerular filtration rate. Let me say, where does atrial natriuretic peptide comes from? Well, the name kind of tells us. So we're dealing with the atria of the heart. So ANP is going to be secreted when we detect overstretch of the cardiac atria. Our renin-angiotensin-aldosterone system. So we've talked about this when we did cardiovascular. We talked about drugs like ACE blockers or ACE inhibitors and beta blockers to reduce and lower blood pressure. So we could run through this scenario and see exactly what's going on. We start with a disorder, an imbalance. So we start right at number 1, where we have dehydration, sodium deficiency, and or hemorrhaging. What results is a decrease in blood volume. That decrease in blood volume is going to be detected by the juxtaglomerular cells of the kidney, which is going to in turn stimulate a release of renin. Now, concurrently, the liver is going to be producing this enzyme called angiotensinogen, and angiotensinogen is going to help to increase angiotensin 1, which is then going to make its way over to the lungs, and the lungs is going to produce an enzyme called angiotensin converting enzyme, and this is going to convert angiotensin 1 into its active state of angiotensin 2. Now, if we take a step back and go back to ACE inhibiting enzymes, we talked about people with high blood pressure or chronic hypertension tend to get certain drugs like beta blockers and, more importantly, ACE inhibiting enzymes, ACE inhibitors. What this does is it blocks ACE, which ultimately stops this pathway right here, stopping the conversion of angiotensin 1 to angiotensin 2. Now, we'll come back to this, but let's follow this route. So let's erase what we did here and assume everything is normal, no hypertension. We continue to angiotensin 2, which goes up to our arterioles and is going to vasoconstrict. So it's going to vasoconstrict those arterioles, which will ultimately lead us to an increase in blood pressure until we get to a normal level of hydration. So again, let's go back to our ACE inhibitors. We block ACE, which blocks angiotensin 2, which blocks vasoconstriction of arterioles, which does not increase blood pressure. Okay, so let's go get rid of our clinical connection here and start looking at some other scenarios, other players here. So angiotensin 2 will also communicate right here with the adrenal cortex of our adrenal gland, which will stimulate an increase in aldosterone. And if you remember, aldosterone played a key role in sodium and potassium secretion and reabsorption. So we're dealing with the kidneys again. So aldosterone is going to work with the kidneys. It's going to increase sodium and water reabsorption and increase secretion of potassium and hydrogen ions. And then as a result, by doing this, we increase blood volume, which increases blood pressure until we return back to normal circumstances. The adrenal cortex, if we look right here, okay, we're going to detect an increase in potassium in the extracellular fluid, which will also kick off our aldosterone release, which kicks off our renin, angiotensin, aldosterone system. Table 26.2 summates all parts of glomerular filtration rate and the regulation of. So we can look at each and every one of the regulation, the stimulus that causes it, and the mechanism or the site of action and its effect on GFR. So first and foremost, I want you to go all the way to the effects on GFR and notice the trend. What we're doing is mostly decreasing GFR as a result of too high of a blood pressure being detected. In one instance, we are increasing GFR here due to another issue with the atria overstretching. So let's look at these mechanisms. Myogenic mechanism. So what's the stimuli? Increased stretching of smooth muscle fibers in that afferent arteriole due to high or chronic hypertension or high blood pressure. As a result, the stretched smooth muscle fibers contract, narrowing the lumen of the afferent arteriole, decreasing GFR. That tubuloglomerular feedback, so we're detecting too much sodium chloride at the macula densa. This is due, in fact, to high systemic blood pressure. Think back to the I Love Lucy scenario, where that conveyor belt is moving way too quickly. So too high of blood pressure, get too much sodium chloride moving through. What happens? We decrease the release of nitric oxides, those vasodilators, by the juxtaglomerular apparatus, causing constriction of the afferent arteriole. Overall net effect, decreasing GFR. Our neural regulation, increase in activity level of the renal sympathetic nerves, release norepinephrine. This is going to constrict the afferent arterioles through activation of our alpha-2 receptors, and we're going to increase renin release. Hormone regulation, here's our angiotensin 2. Decreased blood volume has been detected, or blood pressure, stimulate production of angiotensin 2. As a result, we're constricting afferent and efferent, so that's one of our differences, afferent and efferent arterioles, giving us a decrease in GFR. Here's our one outlier, AMP, atrial natriuretic peptide, detects stretching of the atria in the heart, is going to stimulate secretion of AMP. What happens? We're going to relax those mesangial cells in the glomerulus, increasing capillary surface area, which ultimately gives us our one increase in GFR. Thank you for listening. I hope you found it helpful.